The overall goal of this procedure is to generate human or mouse cardiac progenitor cells from embryonic stem cells. This is accomplished by first culturing high-quality embryonic stem cells, or ESCs, on mouse embryonic feeder cells, or MEFs. In the second step, the cells are transferred to gelatin-coated petri dishes to facilitate the formation of the first embryoid bodies. Next, the cells are dissociated and growth factors are added to the cultures to induce the second form embryoid bodies. In the final step, the embryoid bodies are dissociated again, and the resulting cells are treated with additional growth factors. Ultimately, the cardiac progenitor cells can be further cultured for cardiomyocyte and smooth muscle cell differentiation.
While attempting this procedure, it is important to test the differentiation efficiency of each cell line to allow the selection of the cells with the best cardiogenic potential. Now, Dr. Ieng Lam, a postdoc fellow in my laboratory, will demonstrate the procedure.
To induce cardiac progenitor cell differentiation from mouse embryoid bodies, begin by culturing mouse ESCs on MEFs in ESC medium until the cells are 90% confluent. Next, wash the cells in DPBS and then detach them with 0.4 milliliters of 0.25% trypsin at 37 degrees Celsius and 5% carbon dioxide. After 5 minutes, add 1 milliliter of pre-warmed medium to halt the enzymatic reaction. Spin down the cells for 5 minutes at 200 times G and room temperature, and then resuspend the cells in 1 milliliter of ESC medium. After count